All right, folks. So I see some of you've taken the microbial ecology exam. It's looking good. Make sure that you do that. I think the 24th was the deadline on it, so we don't run into issues with having to reopen it and all of that. We're going to go ahead and jump into the last unit, and this test will be open for that last basically week of class. I think from May 1st through 5th, something like that. So that'll be it, So anyway, I forgot to look up exact dates of when everything's got to be in and all of that, but basically you'll have right up to the end essentially to do that. So I'll let you know when to expect that exam and when it's going to go down and all that business later. But let's get into this. So the last two things we're going to talk about are in chapters 11 and 12, and they deal with controlling microbes. Right? So one reason we want to do that is to prevent infection, right? So you can control them, limit them, and so forth before the infection actually arises. Then, if somebody does get sick, what do you do then? And we're going to talk about antimicrobials and how some of those function. We're not going to get way deep into that with regard to antifungal and even antiviral stuff. It's mainly going to be about antibacterials, all we'll have time for. I may include a little bit on some of those things. It is sort of timely, for example, to talk about antivirals. There's a lot of misconceptions about those. So might include a little bit more on that than I normally would have. So be looking for that. Okay, so let's get started on this. Go ahead and jump into this chapter. This is physical and chemical agents for microbial control. Talk about those. All right, and so physical and chemical, and they also recognize what are called mechanical methods. I guess those technically are a form of physical method, but mechanical means of removing microbes is literally to move them. To physically move them from one place to another, as opposed to doing anything to kill them and so forth. So, grubbing, washing with a wash rag or something like that would be an example of a mechanical method for limiting the amount of microbes in a given area. So it's really pretty simple, but can be pretty efficacious if it's done in the appropriate context. So. What we want to do, of course, is prevent infection, and we also want to prevent spoilage. We don't want to lose valuable food stuffs, whatever those might be, to the organisms that could contaminate them and cause them to spoil. So we want to limit that. So these are techniques for doing those kinds of things. And here then are a list of most of the things that we would be considering when we're going to apply these techniques. So any sort of prokaryotic or eukaryotic form that is microbial that could make you sick or that could spoil food are things that we would be interested in limiting. And so this little flow diagram here, this branching dichotomous sort of diagram has under this one umbrella here microbial control methods, the three different categories that we recognize: physical, mechanical, and chemical. And under those, it shows the different subcategories that exist. And you can see designations such as disinfection versus antisepsis versus sterilization. Now, those are the various possible outcomes that can be obtained utilizing these different approaches. Down here it defines those terms, but what is meant by disinfection, sterilization, and antisepsis. So if you're disinfecting, and by the way, people misuse these terms all over the place, right? We especially misuse sterilization. Like, for example, when you take, say, a baby bottle, like the plastic nipple on the bottle, one thing that parents do is try to clean those assiduously so that their kids don't get sick. And so they might occasionally put them in boiling water. And, the claim is that they've sterilized them. Well, as we'll see, that doesn't sterilize, right? We're misusing the term sterilize. If you were to, say, take an alcohol little pad or cotton ball with alcohol and clean an area on your skin, say prior to hypodermic injection. Well, some people will claim, well, I'm sterilizing this place. No, you're not. You're not sterilizing. You're actually performing antisepsis. So know the difference. So disinfection involves destroying or removing microbes from inanimate surfaces. So there's our operative word. There's the key word. It's inanimate. That means not living. So a coffee table, a lab bench, a glass that you would drink out of, things like that. Your cell phone. If you were to try to reduce the load of microbes on those using some physical, chemical, or mechanical approach, you'd be performing disinfection. And antisepsis is sort of the antithesis. It's the opposite of disinfection in that it is used to destroy or inhibit pathogens on body surfaces. Okay, so on mucous membranes and on skin in particular, that's where we would apply antisepsis. And notice it uses the term here, vegetative pathogens. Remember, those are distinct from endospores. If you're going to kill endospores, the various approaches that we have that are the usual means of antisepsis won't do that. All right, they just won't because they're just too hardy. They're too resistant. And so, if we're going to kill endospores, we're going to have to go to the extent that we would actually be harming the body. We would be doing more harm than good. So antisepsis, when you use an alcohol, say prep pad, to clean an area, in somebody's cubital region for hypodermic injection or something such as that, that can only be applied to surfaces that can handle that. Sterilization is where we go to the extent that we kill everything, right? Everything. Nothing's living. If you've sterilized some glassware that you're going to use in a lab, put it into an autoclave, set that appropriately, and we'll talk about what those settings are later, but you'll set it appropriately, turn it on and run it to complete the cycle. When things come out of there, initially they are sterile. There's nothing living on them. And you can, cover them and protect them in such a way that they'll remain sterile even after they come out of the autoclave. Some things can be made so that they remain sterile after that until, you open up the container that they're in. Obviously, if you let air in, there are spores, there are individual microbial cells, there are droplets, there are aerosols containing organisms of all kinds, and even some non-living microbes like viruses. Those are going to get on those surfaces. It just stands to reason that that's going to happen. So, but sterilization is possible. We've got to go to fairly extreme lengths to do that, but it is possible, and it's only possible on inanimate objects. We're not going to sterilize somebody's body anyway. It's just not going to happen. So now you can see that most of these techniques do have as endpoints the possibility of either sterilization or disinfection. Antisepsis is pretty much only limited in this case to the outcome of applying some liquid agent on animate surfaces. But everything else that's up here that we'll be talking about can be typically applied in such a way as to either achieve disinfection or, if we go further with it, sterilization. Anyway, so these are the things we're going to talk about: incineration, use of dry ovens, steam under pressure. That's an autoclave or a pressure cooker. Using boiling water, pasteurization, filtration, running air or liquids through filters, use of a variety of gases and liquids. So the choice that we go with in the end is going to be in part dictated by the kinds of organisms that could potentially exist in a given context. And so if you're working in a healthcare setting and your goal is to prevent infection basically at all costs, you will assume that there could potentially be the highest resistance kinds of microbes out there, and you will do whatever you can to prevent their occurrence so that they don't make their way into bodies and cause infection. Okay? Whereas if you were working in another setting, which could relax some of the constraints a little bit, you might make the assumption that mainly what you've got are these organisms of least resistance. And if you go to the extent that you can take those out, and then it will be good enough, right? That it'll be adequate to more or less maintain everyone's health and reduce the likelihood of sickness or spoilage. And then you've got these moderately resistant forms, which likewise are things that if you're a healthcare person, you would probably really want to limit. And if there's any potential of them occurring in other settings, you might think about limiting these as well. So the high resistance forms are going to take a whole lot of work to try to get rid of. Moderately resistant forms will take quite a bit, but not as much as the high resistant forms. And then the least resistant forms are the things that you can destroy fairly easily with standard methods that are available to just about anybody. You can go to the grocery store, you can go to Kroger or Walmart and buy cleaners, disinfectants, and things that will be operative against these. But do know that not all organisms are created equal. And notice here, under the least resistant heading, we have enveloped viruses, all right? And the coronaviruses that, the one we're dealing with now, the new SARS coronavirus 2, plus those that have plagued us in the past, like SARS coronavirus number 1, MERS, and other enveloped viruses include influenza and so on. They all have an envelope of phospholipid, and it turns out that that's actually relatively easy to disrupt with things that are going to act as surfactants that will break up that phospholipid bilayer. That's broken up. The spikes that are in there that the virus uses to attach to receptors in a host cell membrane, are going to be essentially diffused. They're not going to work properly. And so envelope viruses, in the grand scheme of things, are not all that difficult to destroy. All right. But you have to take the measures to do the destruction. And you have to be aware that they could possibly be present. Right? So sometimes those criteria aren't met, and you wind up having these viruses hang around for a while, and people get sick. So Anyway, there's variety among organisms in terms of how resistant they are to our methods to control them. And so, um, one of the things that sort of brought out by that coverage of organisms, microbes, and their variation in resistance has to do with the comparison between endospores and the vegetative cells that could potentially sporulate to give rise to those. A nice point of comparison is to look at that. So remember, these are the forms that we've talked about before, the gram-positive rods in the genus Bacillus species, and Clostridium. Those two genera in particular are among those that are most well known with regard to the ability to sporulate, the gram-positive rods. And so if we were to take, say, for example, Bacillus anthracis and look at it in its vegetative form and then compare that against the endospore form in terms of how easy it is to destroy them, this is what you would get. So you could apply moist heat. Okay? That's water vapor or boiling water. If you do that and you get your temperature up to 80 degrees centigrade, that's not even boiling. That's very hot. It's enough to do some damage to your skin if you were to get it on you. It's not quite boiling. It turns out that for vegetative forms of the bacteria, that's adequate to kill them, right? But as we know with Bacillus, and we've seen this abundantly in the lab whenever we did endospore staining with live cultures of Bacillus species, I think we used Bacillus subtilis this time around. There are ubiquitous spores, even in relatively youthful cultures. So you can kill all the vegetative forms all you want, but if there's endospores present there, that level of heat, that level of moist heat, is going to be inadequate to kill the endospores. So let's say you had a test tube of nutrient broth and you're growing Bacillus species, whatever, Megatherium, Cetulus, Anthracis, doesn't matter. Let's say you go ahead and you boil that tube of turbid bacterial broth and you kill all the vegetative forms in there. And then you go ahead and take that same tube and put it into an incubator and allow it time to rest. The organisms will start growing again. You could also take a sample of it and put it onto some sterile nutrient agar. You'll get growth, even though you've killed all the vegetative forms. Why? Well, there were some endospores left behind. Now, if you want to kill everything, if you want to make it so that tube is done and there's no way it can actually begin to recover, you'll heat that tube all the way to 120 degrees centigrade. Now, that's pretty striking because normally water, and this we're talking about here is water vapor, it will only reach 100 degrees centigrade. That's all the higher the temperature can be under normal conditions, right? In a laboratory. But if you want to go higher than that, you have to increase the pressure. That's what an autoclave does, and that's what a pressure cooker does, the same thing. So that level there is what's obtained in a pressure cooker or an autoclave, and that's adequate to kill them. So when we're talking about the difference between endospores and vegetative forms relative to their ability to resist death by moist heat, these data indicate that if you take 120 and divide it by 80, that gives us a measure of the level of resistance of endospores in comparison to vegetative forms. Endospores are 1.5 times more resistant relative to the vegetative forms. And then you can see for these other factors like radiation, applying something that is ionizing in its effect, like X-ray radiation, destroys DNA, so it's going to destroy cells. You can see endospores are more resistant to that than vegetative forms are. So there's 4 times the resistance of endospores relative to their vegetative counterparts. And sterilizing gases, you can come up with a device that create this gas of things that are toxic to bacteria and more or less flood them with that. And you can see ethylene oxide, also known as ETO, was the substance used here. And again, endospores are more resistant to that than the vegetative forms. And finally, you can use a sterilizing agent like glutaraldehyde, similar to formaldehyde, but a little better actually. It is a sterilant if it's applied properly. And you can see there it takes 3 hours of application of glutaraldehyde to get complete death of endospores as opposed to just 10 minutes for the vegetative form. There's an 18 times difference. Right? So this hopefully points up the fact that endospores are really challenging. And if your goal is to sterilize, you're going to have to put effort into that and use special approaches in most cases. Whereas here with the vegetative forms, if it's okay that you just limit your efforts to killing those, then you can see where you can get by with a little less. All right, so there's that. All right, so yeah, I'm throwing these terms around: sterilization, disinfection, antisepsis, sepsis, sanitization, and degermation. Those mean, look for operative keywords in all of these. So in the case of sterilization, the word all, all viable microbes, including viruses and endospores, everything. And in fact, we should include in there prions as well. The assumption when we sterilize is that we've taken those out as well. If we've applied our techniques properly, we're likely to be able to achieve that. Sterilization kills everything, destroys everything. Disinfection, okay, the key word here, well, a couple of them. It destroys vegetative pathogens, but not the endospores, and we apply this on inanimate surfaces. That means non-living. So a window, a tabletop, a fork, your car steering wheel, all of those things can be disinfected. Antisepsis, that's use of the same kinds of substances or similar substances to those that we would use in disinfection. But here we're only able to remove microbes from exposed body surfaces. And technically, the oral cavity, the buccal cavity, and the pharynx would qualify. Those are places that we can apply antisepsis. Elsewhere, it's really sketchy because you're liable to damage some very delicate surfaces and other tissues in a way that could ultimately harm the individual's ability to thrive. So antisepsis is only the skin and mucous membranes, principally in the mouth. And then we've got sanitization. Now, sanitization and degermation are linked in that they're both mechanical methods for removal of microbes. All right. So that means using some sort of force, some sort of friction, some sort of scrubbing action, a scrub brush, a rag, or something like that, and rubbing something down. A dishwasher would be an example, because you're using water there, jets of water, and you're spraying them, and you're striking surfaces, and you're knocking stuff away, and rid of them. And that's preferable, I think, in the case of, like, say, sanitization in the dishwasher is preferable to um, other techniques for making, forks and spoons and knives and glasses and so on usable. Um, Consider what would happen if you didn't mechanically remove, stuff that's on the plates and on the the silverware that you would use after you eat. Let's say you just throw all that stuff on them, all that debris on them into an autoclave, and you sterilize, right? You can sterilize the plate with, a bunch of grease and bits of food and stuff like that. And then, so it's sterilized, right? You take it out of the autoclave and you set it down. Are you going to want to eat off of that? Ask yourself that. No, there's a bunch of junk on there. It's devoid of organisms, but it's little bits and pieces of leftover food that somebody else left on there. So sanitization, and maybe even applying that with some disinfection too, is preferable, because it removes the unsightly little things that are left behind. All right, so yeah, cleaning an inanimate surface mechanically, that's sanitization. If you were to take like a greasy roasting pan after, say, having an Easter ham or something like that, and take some sort of a stiff bristled brush and start scrubbing on that, knocking away the material in the bottom of it, you're applying sanitization essentially. Degermation is the same thing, but it's just your skin. If we were to take some little, say, um, uh, sort of pad that is sort of scratchy a little bit, they make those little buff pads or whatever for your skin to remove, dead skin and that sort of thing. If you were to apply that with just some hot water, for example, and just kind of scrub a surface of your skin that way, your face or something, you'd actually be doing some degermation. Now, normally we would apply some soap with that. The soap may actually give us some antiseptic effect, right? But if you were to just use a wash rag or some sort of a scrubber devoid of anything on that chemical, in a chemical sense, you'd be applying pure degermation. And that's adequate sometimes. It's maybe all you need in some cases to limit the occurrence of microbes that could cause sickness or other things. Well, be aware of the variation in these terms and what they mean, and learn how to use them accurately, correctly. So yeah, this little table here is a good one to look at because it gives definitions as well as examples. Right? And so, when we apply these techniques, besides knowing what the potential organisms are, level of resistance, it may also help to know the factors that affect death rates of microbes. That can dictate how exactly we go about implementing different techniques. So, and how much we've got to do. So if you've got a really obviously, maybe even visibly contaminated surface where the number of microbes is, astronomical, that's obviously going to take a lot more work than if you were to have lower numbers of microbes to deal with. So that's something that could bear on your decision about how to go about doing your work. The possible nature of microbes in a population, if you're in a healthcare setting, and there's the possibility of things like, pseudomonads and Mycobacterium species, the Mycobacterium tuberculosis for sure, Clostridium species, and so on. That's going to require more elevated effort than would be the case, say, in your own home, where the likelihood of those organisms is much, much less. The temperature and the pH of the environment, okay, so that's going to affect how readily things die. a particular disinfectant or antiseptic may only be able to do an effective job of killing microbes when the temperature is within some narrow range, say 25 to 35 degrees centigrade, which is normally around the temperatures that we operate at in our daily lives. But if, say, the temperature were to be elevated too much, um, it may reduce the effectiveness of the chemicals and your antiseptic or your disinfectant. And likewise, really cold temperatures can limit the effectiveness of um, chemical or other agents that could um, destroy organisms. So you got to think about that. Is the temperature of the environment or the pH, the acidity or alkalinity of the environment, are those adequate for utilizing this approach? And if they're not, then trying to find some ways to correct that may be in order. Of course, concentration or dosage of the agent. You use too little, you can maybe put a dent in the microbial population or the community size, but it's probably ultimately going to be ineffective. So if you can help it, you want to kill everything, just so as not to leave behind survivors that maybe survive because they had a little bit of resistance to whatever you're applying. And so if they survive and they're genetically sort of given that capability, then when they reproduce, that's going to lead to a shift in the population toward more and more individuals being resistant. Ultimately, you'll have a big problem on your hands, much the same way as we've seen with antibiotic resistance. So there, with antibiotics, people will take them, but they don't take the whole bottle, and they more or less promote the evolution of antibiotic resistance. Well, the same kind of thing can happen with disinfectants and antiseptics and so forth. So the amount of the agent is important. In most cases, too little is the problem, as opposed to too much. Mode of action of the agent. So if you're trying to get organisms removed from a surface that's got a lot of oil and other kinds of dirt on it, that might be something that limits the ability of the agent to get into the organisms that are actually potentially destructive, potentially infectious. So you want an agent that will cut through the grease and thereby be able to enter the microbial envelope and go in and kill those organisms. If you apply something that is, say, not able to get through a greasy buildup, something that is hydrophobic, it's just not going to work. So, and that kind of ties in with this down here. The presence of different things in the environment that can limit the effectiveness of microbes needs to be taken into account. Dirt, things like fecal material or urine or blood, other things like that. If they're present in a setting, and there's a lot of those things, that can greatly limit the effectiveness of disinfectants and sterilants and that sort of thing. So you'll have to elevate the intensity with which you apply your disinfection or your antisepsis or what have you. Anyway, those are all factors that can make the difference. Recognizing these and making accommodations for them can make the difference between a failed attempt at disinfection versus a successful one. All right, so what I always do when I get to this section is I talk about, how a student might think of themselves as being someone who's been recently hired to develop disinfection or antisepsis techniques. And so if you put yourself in those shoes and you think about, okay, what would that entail? The first thing I would say is think about your enemy. Think about who it is that you're trying to limit. What are their weak points? What are their vulnerabilities? How do we exploit those? All right. So you can brainstorm on that. Right? And you know now, for example, with bacteria and other prokaryotes, their basic makeup. All right. And so you might be able to think of ways in which all of those parts are potentially vulnerable. Okay. What are the chinks in the armor, if you will, of different microbes? So with regard to the prokaryotes, bacteria, not archaea because they're not pathogenic, but pathogenic bacteria, they all have cell walls. And fungi have cell walls. Don't forget that. So if we're worried about fungal infections, this is something. Yeah, so the cell wall, that's on the outside of the cell, all the way on the outside of the cell. The only thing that lies beyond that would be the glycocalyx. And so obviously, if you were to disrupt that, you're going to disrupt cell function. All right, it's a very important component of the cell. So you could target that and have some means by which you can break it up or weaken it or make it fragile, as it says here. And a lot of physical and chemical agents work that way. Ultimately, what's that going to do? Well, it's going to limit the organism's ability to maintain homeostasis, like ability to regulate all the things it needs to regulate going on in the cell, and it's going to fail. So the cell will lyse. It'll essentially explode. So yeah, the things that can do that: detergents, alcohols. Detergents would be especially good, and alcohols would be especially good on gram-negative cell walls because they contain, remember this LPS, right? Lipopolysaccharide, also known as simply the outer membrane, also known as endotoxin. Remember that? Hope so. All right, that's lipid. It contains lipid. And what do detergents do? What do alcohols do? Well, they break up lipid. All right, so if you apply those and you've got gram-negative bacteria around, that LPS layer is going to fall apart, going to be disrupted, it's going to be compromised, and those bacteria are going to be likely to cause infection. That's good news because a lot of those gram-negatives are bad. And there are some antimicrobial drugs that do the same thing. Some things we're going to talk about in Chapter 12 that do a similar kind of thing. All right, so there's that. That's a point of vulnerability. If you can attack that, great. The good thing about the cell wall is humans don't have cell walls. So none of these approaches are going to affect our cells all that much. They'll affect the cells of bacteria and other things that have outer coverings with some sort of lipid in them, like an envelope virus. Okay, another point of vulnerability. Let's work our way in. So beyond the cell wall, or just inside of the cell wall, we have the cell membrane. The cell membrane. I don't know. This down here, surfactants shouldn't be part of that. It got highlighted somehow. But yeah, the cell membrane can be compromised. It can be attacked. And the way you do that is with detergents and surfactants. Really kind of redundant because detergents and surfactants are the same thing. You disrupt the cell membrane, remember this is the regulatory sort of entry point for things coming and going from a cell. So if you disrupt that cell membrane, again, the cell cannot regulate the difference between what's on the outside and what's on the inside. And that's how cells work, is they set themselves apart from everything else. So the cell would then maybe not be able to keep out extra water. If that's coming in, the cell's going to lose that ability to flush it back out. May not be able to secrete substances as readily as it did. Sometimes cells need to do that. all of those things that could disrupt its integrity. So attacking the cell membrane is another way to go. Very vital part of the organism. So detergents slash surfactants, whatever they might be, alcohols, soaps of different kinds, they work this way. So here is a phospholipid bilayer, these little beads with the little tails, the fatty acid tails. And these little things here are supposed to be representing surfactants. So when you apply those, they have chemical properties much like those of the phospholipids in the membrane. And so they have, they're not antipathic to one another. They like each other, essentially. And because of that, then these surfactants can get in here and literally try to join up with that membrane. Well, in doing that, they break it apart, all right? And you can see ultimately what that means is the integrity of that membrane is so disrupted that the cytoplasm oozes out and materials on the outside make their way in. That cell is going to undergo destruction or lysis, all right? That's what happens there. That's a cell that's not going to make it. So that's how surfactants work, slash detergents, if you want to call them that, that's fine. They break up the surface tension afforded by what normally would be this attraction between these phospholipids. All right, break that. They disrupt that. All right, so those are on the outside. Cell walls and cell membranes. What are some things you can target on the inside? Well, you can go in there, and one of the main things, of course, that keeps that cell going would be its genetic material and the enzymes and other proteins that are coded by that genetic material. So disrupting either one of those will usually disrupt the other. All right. So if you can, say, knock out protein synthesis, many of the enzymes that the organism, all of the enzymes that the organism is using are proteins. Those same enzymes, some of them, might be needed for nucleic acid synthesis. So you knock out the protein, you're going to knock out the nucleic acid synthesis, and vice versa. If you were to disrupt, DNA function, transcription and translation, you're going to knock out protein synthesis. So those go hand in hand, and it's arguable that those are really the essences of life. That's what life revolves around, are those two things. So preventing replication, transcription, and translation of DNA, that shuts down heredity, and that cell is not going to make it. It's not going to be viable. Disrupting peptide bond formation, that keeps proteins from forming. Peptide bonds occur up between amino acids. If you can stop those bonds from forming, no proteins are ever going to form. And then protein synthesis, disrupting other aspects of that can also be helpful, weapons to use against bacteria. So it mentions the different things that can do that. They give an incomplete list of things, but chloramphenicol is an antibiotic substance we'll be talking about in Chapter 12. A very dangerous one, I might add. That's used to treat sort of systemic infections in bone and that sort of thing that could spread to other parts of the body and cause serious consequences, could cause sepsis. Kind of an antibiotic of last resort in many cases. But using different kinds of radiation like UV radiation and using formaldehyde, those are all ways to disrupt those things. Now, the problem here is, as I said with chloramphenicol, these are dangerous, not only to the organisms that we're applying them to, but to ourselves as well. So if we're going to use these approaches to target organisms, we're going to have some protective equipment. We're going to have to apply them in a way that keeps it from backfiring on us. So there's that. So you can disrupt protein synthesis, nucleic acid synthesis, and then Another approach, sort of the last one that you might envision, is to disrupt the proteins themselves. So if they're already present, you can't disrupt synthesis of them. So the next best thing would be to disrupt proteins while they're active, while they're in place, and that could be effective as well. All right. So, because remember, proteins are enzymes. All of the enzymes which occur in a cell, these catalyze all the biochemical reactions, of which there's at least several thousand in most instances. And then you got structural proteins. So that would be things like cytoskeleton, proteins in the flagellum, spikes in the cell wall, say of a gram-negative organism. They could have little proteinaceous sort of spike-like structures there. Various things that are proteinaceous that are not used specifically as enzymes, but are helpful in other ways. If you could disrupt those, you're going to disrupt back microbes. All right, so this picture here, I'll go through this, and then I'm going to stop the lecture so that you can digest that, and then come back at you with a second and possibly third lecture on this, and finish it off. Yeah, so this shows with regard to proteins, which are incredibly important molecules, cells, to keep them functioning. This is the shape of, say, an enzyme that is functional. That's shaped in such a way to properly accept substrates. The substrates are the chemical substances that are bound in particular places on that enzyme. They're shaped and they're charged properly to hold those things, and those substrates in turn will be used as reactants in biochemical reactions. So substrates could include, say, carbohydrates that have been taken in for energy. An enzyme will hold that carbohydrate, say that glucose or whatever it is, and present it in such a way that it can be acted on by other substances and broken down and energy released from it. So enzymes are chemical helpers that catalyze reactions, and they have to be shaped properly, right? What they are strings of amino acids, bent and folded and twisted and contorted to arrive at these sort of three-dimensional shapes. So that's the native state of the enzyme. And you can see also in here they show these little dashed lines. All right, those are like weak chemical bonds, hydrogen bonds, and other kinds of linkages that are holding that shape, maintaining that shape. All right. Now, if you apply some heat, it's a little bit of heat. Sometimes it's all that's needed. Higher than would be the normal operating temperature for the organism. Let's say it's a mesophile that functions best right around the temperature of the human body. And you elevate that temperature up, to say, instead of 90, 98, 99 degrees Fahrenheit, you elevate it to 110, 115, 120 or higher. You'll likely disrupt that protein, and it won't function like it formerly did. You can see how just application of a small amount of heat has, if you look at the shape of this blue enzyme down here, it's the same enzyme, all right? It's the same one as the purple one up there, but these little bonds that put tension in this and hold it in that particular shape have been changed, have been altered. So this active site is lost, and the one that formerly existed right around in here is lost. That enzyme can't function. Now, if we apply even more heat, we go to this extent, and we basically disrupt the whole thing and it falls apart. But once that happens, we've achieved what's called denaturation. Now, I'll tell you, if you were to remove the heat, and expect the protein to go back to its native state, I'll put a big X through that arrow. That's not going to happen. All right. It's not reversible. So denaturation is irreversible. Let's write that word. It means you can't go back. All right. So that's complete and total destruction. Obviously, if that's an enzyme the organism needs, it's not going to make it. It's going to die. All right. So now notice also the pH changes in here. pH has to do with the concentration of these things. Hydrogen ions. It's a hydrogen ion. A hydrogen atom that has lost an electron, which literally means all that's left of it is a proton. So substances that give off lots of those in solution, you mix them with water and they release a lot of those, that increases the acidity of that solution. So the more of that stuff there is, the more acid it is. The other sort of way to go is to solubilize things that then break down to give rise to OH functional groups. Hydroxyl, called a hydroxyl ion or hydroxide ion. Hydroxide, not hydroxyl, excuse me. So if you elevate that, that'll change the pH. That'll make it go way up. Okay? So hydrogen ion brings the pH down. Hydroxide takes it up. Either way, you can disrupt proteins. Right? And that'll change their shape in exactly the same way as heat will. So there's that. And then finally, in this diagram, it shows another thing that can happen with enzymes is they can bind in these active sites, the places that normally bind the substrate. They can bind a little collections of atoms of different substances, and those then will block that site. And while the enzyme itself hasn't changed its shape, it still is inactivated. It's not going to do what it should do. Because substrate, if you have substrate, and it comes along and wants to get in here, well, that can't happen. It's going to block it. So this is called, and it's a little bit enigmatic. I'm not exactly sure why they came up with this name, but this is called, let me erase, not an M, that's an N. This is called oligodynamic effect. Right? An oligodynamic effect, and the thing that can achieve this would be use of some sort of a heavy metal. Those would include things like lead. You write it over here, lead, arsenic. What else? Selenium, I think, is one. I don't know, mercury. A lot of things that are pretty dangerous to humans. If we can find a way to apply those, though, in a way that keeps us safe, it can actually destroy microbes that could cause infection or spoilage. So that's not as commonly implemented these days, but that's another approach, is to use heavy metals for this purpose. Okay, so if you wipe your hands with an antimicrobial cleaner, you are accomplishing which of these? So is it sterilization? Well, no. Body surfaces can't really be sterilized. You'd have to kill everything, and doing that would not be possible without doing grievous, extreme, irreversible harm to your hands. So no, we're not going to sterilize. Is it disinfection? Well, disinfection isn't quite the same as sterilization. It's killing some organisms, not all. But disinfection is applied to inanimate surfaces. So your hands are definitely animate. I hope so, anyway. And then antisepsis. That deals with animate surfaces, the skin, that's a check. And it is a means by which we use cleaners, antimicrobial cleaners, right? Um, yeah, that one is going to work. So C works. Now, sanitization, what's that? Well, that is mechanically removing debris and organisms could be mixed in with it off of inanimate surfaces. So, like forks and spoons in a dishwasher, that would be sanitization. So, yeah, the answer here is C. Right? Antisepsis. All right, so I'm going to stop it there, and I'll come back at you with another lecture here shortly. All right.